Okay. It's here. Okay. Thank you all for participating in today's media briefing. I'm joined by Dr. Yi Shi, Assistant Professor of Cell Biology at the University of Pittsburgh School of Medicine, and Dr. Paul Dupré, Director of Pitt Center for Vaccine Research. They're going to share a major scientific advance developed in Dr. Shi's lab that moves us toward a potential new drug to treat and prevent COVID-19. After our experts give their remarks, we will take your questions. First, I'd like to play a short video that summarizes the project.
White, and it turns out that the first time I saw Wally's picture was really interesting because it's a little bit reminiscent of my black Labrador Wally. That's why we named him Wally the llama. We inject llama Wally using the virus spike protein, and after 50 to 55 days, we took the blood from llama Wally, and then we used this technology we developed using mass spectrometry to enable large-scale discovery of the potent neutralizing nanobodies from llama. So humans produce antibodies when they encounter or infected by the viruses to neutralize the virus. And the nanobodies are very small antibodies produced by llama that can do the same job. And they're extremely potent and stable, which can be used for therapeutics. Compared to the monoclonal antibodies that usually requires intravenous injection, our nanobodies allow the possibility of direct inhale, which makes rapid delivery of the nanobody to the major infected organ, which is the lung. We're currently collaborating with the Center for Vaccine Research at the University of Pittsburgh to perform preclinical experiments using different SARS-CoV-2 animal models. And I can tell you that we get very encouraging results.
Hi. So for most of the mammals, including with humans, we can produce monoclonal antibodies. However, a llama, alpaca, and camels can naturally produce single domain antibodies. And the warhead of these single domain antibodies are called nanobodies. That the small size of these nanobodies is highly advantageous for drug delivery. And we have discovered so far the most potent neutralizing nanobodies basically in the fight against COVID-19. So the idea of using llama nanobodies for drug discovery isn't really new or novel, but they're very difficult to find with the conventional technologies. And since the pandemic began, the few SARS-CoV-2 nanobodies reported have not been terribly effective at neutralizing the virus. So in my group, for the past 3 years, we have really been trying to develop powerful technologies to enable high throughput nanobody discovery. So we can identify thousands to tens of thousands of very high affinity nanobody targeting literally any antigen of interest. So using this technology very quickly, within 3 months, we discover some of the most potent nanobodies that can neutralize SARS-CoV-2 virus. So today, we published a paper in the journal Science magazine describing SARS-CoV-2 nanobodies that are orders of magnitude more powerful than anything previously reported. So with the help of the collaborator, Dr. Cheng Zhang at the University of Pittsburgh and Dr. Dina Schneidman at the Hebrew University of Jerusalem, we performed structural analysis and observed the wealth of diversity in the ways that our nanobodies can bind and attack the virus spike protein. So showing you here just a little bit of one of the most potent bioengineered nanobodies in complex with the virus spike, and you can see how this beautiful structure work. So basically, in this particular case, the nanobodies will sit on top of the virus and will block the virus from opening, or the virus spike from opening to bind the human receptors for host interactions. So that means we can link several different flavors of the COVID nanobodies that we developed together, like a Swiss Army knife, so that if the virus mutate to escape one nanobody, another will be there to keep it in check. So our nanobodies are extremely stable. They can sit at room temperature for up to more than 6 weeks, if not longer. And they could be freeze-dried for shipping and for storage, which is really important for the drug development. And rather than having to be delivered through a intravenous injection throughout the entire bloodstream, like the monoclonal antibody currently in clinical trials for COVID, our nanobody can be fashioned into an inhaled mist or aerosolization to deliver antiviral therapy directly into the lung where they're mostly needed. So these properties could drive all drive down cost and increase access, making nanobodies much more compatible with the global crisis. So now Dr. Dupre will tell you about the preclinical work the Center for Vaccine Research has done to test how well our nanobodies neutralize the real alive SARS-CoV-2 virus. All right, thank you.
Thank you, Dr. Shea. Back in February, the Center for Vaccine Research here at the University of Pittsburgh was among the first in the nation to receive live samples of SARS coronavirus 2 directly from the Centers for Disease and Control, CDC. We were trusted because we have specialized facilities that allow us to work with this virus, this brand new virus, safely. And that was really important and still is really important because this virus travels through the air. And as yet, we have no safe and effective licensed vaccine for the disease, which is caused by SARS-2, Covid-19. So through our international collaborators, we also acquired samples from Germany. One of the very first cases in Europe was a Munich case, and we were able to obtain that virus. And that's the virus that we've used in this exciting study with Dr. Yi. Since we are virologists, when we got that virus, we quickly figured out how to grow it in our biocontainment laboratory. And we've developed tests to work out how to measure how well vaccines and other therapeutics like these nanobodies work. We can understand, because we have these assays, how to stop the virus infecting cells. What's really nice about the Center for Vaccine Research is we have great neighbors, and Dr. Shea is one of those neighbors. So whenever he needed to test how well his antibodies worked, how well they faced off against SARS coronavirus 2, he naturally talked to us as his neighborly virologists. We work on the same floor. We used to be side by side in the same office location. So we see each other and we chat about science often, and that's how science works, about communicating and talking about new ideas. So we were really excited when we saw the potential of his discovery, and we got to work. We mixed a minuscule amount of his best nanobodies together with that live virus from that Munich patient. And then we added a few drops of that solution to the plate of cells. And eventually, after a couple of days, measured how well those antibodies, those nanobodies, neutralized the virus and how many cells survived. What we found was really quite remarkable. Just a fraction, a minuscule fraction of those nanobodies could neutralize enough particles of SARS coronavirus 2 to protect millions of cells. That's so much more effective than many of the other antibody therapeutics, and certainly the nanobodies that have been identified to date. So we have this exciting paper, pleased to be part of it, collaborating from the Center of Vaccine Research. And now we're exploring how well those nanobodies work, not just in plastic dishes. Of course, that's where you start, but how well they're working in the animal models of COVID disease that we've developed over these many months here in Pittsburgh. And of course, that's for another day. But so far, the results in these models are really very promising. So thank you for listening. And now I think we're going to take some questions.
Thank you both. And now we'll take questions from the media, all of whom are joining us remotely online. To ask a question, you'll have to go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in the queue. I will read your name and your line will be unmuted for you to ask your question. We will try to call on as many different media outlets as we can in the time we have allotted. First up, we have Paul Goff from the Pittsburgh Business Times.
Hi, can you hear me okay?
Yes.
Thanks for taking the time to hold this news conference and answer our questions. I didn't think I'd be writing a story today about llama and nanobodies, but that's awesome. I wanted to check with you guys to talk about what a possible timeline would be on this. What would be the next steps in this clinical trials, and then when do you think it might be put out into the market, as it were?
Yeah, so we're very excited about our discoveries, and we'll try the best we can to move it forward towards clinical trials. But on the other hand, we also will have to make sure that it will be delivered safely to the patients. Thank you.
Yeah, thanks, Paul. That's a good question. As Dr. Shi just mentioned, it has to be delivered safely. And again, just like other therapeutics, it has to go through the normal phase 1, phase 2, phase 3 trials required for licensure of any novel therapeutic. And of course, as you've all learned, that takes time. So what's pretty good about this is it can be bulked up rather quickly. You can make it very effectively, and quite a large amount of it in standard bacterial systems. So being able to ramp up production, one of the big problems with vaccines is really not such a big issue with these nanobodies. They're small, they're producible, they're easy to purify. Whenever we get them for some of the studies that we're taking forward at the moment, they're really very, very clean. And that gives me great confidence looking at them, that expedited processes will be able to move pretty fast forward with those preclinical studies.
All right, next question comes from Tom Averill at the Philadelphia Inquirer.
Hello, thank you so much. I have two questions. One is, I've seen it said that nanobodies from llamas are either a quarter the size of quote unquote normal antibodies, or maybe 1/10 the size. I'm not sure which it is. Maybe it depends on the type. And then the other question is a very non-scientific question. Can you tell us, I apologize, but this is the kind of thing editors want to know. Can you tell us where Wally lives and how you found him and all that kind of thing, and you know, how old he is?
All right, thanks so much for the question. In terms of the size of nanobodies, they're about 1/10 the size of human monoclonal antibodies. So basically, they're about 12 to 15 kilodalton, wherever human monoclonal antibody is about 120 to 150 kilodalton. So they're very tiny, but they're extremely stable. To answer your second question, I think Wally is lives in Massachusetts, in a very nice farm in Massachusetts, and he's a fantastic llama, and just a reminiscence of my black Labrador. And the first time I saw Wally, I was literally shocked because I never saw a black llama, which really surprised me. But yeah, you know, he's currently our best friend. Hopefully, will help us, you know, fight through this disease of COVID pandemic.
Do you know how old he is?
I actually don't know, but I would assume that he's less than 5 years old. Thank you.
All right, our next question comes from Aparna Delani from CBS News Desk.
Hi, thanks for taking my question. I have two questions. One is, what's so special about llamas? Why did you pick llamas and not other animals? And the second one is, what is so different about this study as compared to other studies done on llamas?
Yeah, so I think for most mammals, including us, we can produce regular monoclonal antibodies. As I mentioned, about 150 kilodalton, they have two chains, light chain and heavy chain, and need these two chains to, for example, bind to the pathogens so that we can neutralize the pathogens such as viruses. And in camelid, such as alpaca, llama, and camels, they can produce a special kind of antibodies, a single domain antibody. So they only have a heavy chain and it's devoid of light chain. So nanobodies are basically the warhead of this heavy chain. They're extremely small, as I mentioned, only 1/10 the size of the monoclonal antibodies. And the beauty of these nanobody, that's the reason why my lab is kind of interested, is because they are extremely stable and they're very easy to be produced and with a really low cost, such as they can be produced from the microbes, such as E. coli and E cells, which make them highly compatible with large-scale productions and manufacturers, which are compatible with the current pandemic. So these are the major. And on the other hand, because the stability of these nanobodies make it possible to be directly aerosolized and inhaled to the lung where it's most needed for the neutralization of the virus. So all right, thank you.
All right, the next question comes from Sarah Bowden at WESA.
Hi, can you hear me?
Yes, you can.
Okay, great. So my question, and maybe Dr. Shi already addressed this and I just didn't quite understand. Are humans incapable of producing these nano, I'm blanking on the name, nano antibodies, or do camels just produce, or do llamas just produce them more than humans?
Yeah, so basically humans only produce conventional monoclonal antibodies. They have two chains, wherever llamas can produce, in addition to these conventional antibodies, a special kind of single domain antibodies, as I mentioned, or you can call it nanobodies as a function units of the single domain antibodies. Yeah, so humans do not produce nanobodies, wherever llama are the only known mammalian species that can produce these single domain antibodies. Indeed, you look into the vertebrate kingdom, in addition to camelid, I think sharks can also produce single domain antibody. I think there's a reason why this species, amazing species, can produce this single domain antibody, because they can tolerate some extreme temperature and conditions, such as heat and dehydration, which turn out to be extremely useful for human application, or at least for development of drugs yeah, thank you.
All right, our next question comes from Amy Wattis at KDKA TV.
Hello, everyone. Can you reiterate again how long you've been working on this research? And then my other question, how the drug produced would be administered in humans, and would they only be able to get, would it be several treatments, one treatment, and so on and so forth?
Sorry, the first question is?
Amy, could you repeat your first question?
Yeah, my first question again was, can you reiterate how long you've been working on this research? You may have mentioned it earlier, but I misheard. Right. And then the second one again, how it would be administered in humans.
Sure, yeah, so my lab is actually a very small lab, only 3 people. They're all junior scientists. So basically we spend about 3 years to develop this technology. They enable us to discover and identify drug quality nanobodies. I'm talking about thousands and thousands of these high quality nanobodies. And with this technology, it took us about 2 to 3 months to very quickly discover ultra potent anti-COVID nanobodies. So basically spend a lot of time developing the tools, and when the pandemic hits us, we are working extremely hard and very rapidly generate these ultra potent anti-COVID nanobodies.
And how would they be administered?
Yeah, so there are different ways. I think that's another beauty of nanobodies that allows potential, basically different ways of administration, including inhalation and also the uses as a nasal spray. So this, and potentially you can also use it as an intravenous injection, just like monoclonal antibody. But I think the major advantage is through the nasal spray and also aerosolization, where it can directly deliver to the lung, where it's mostly infected by the virus. Thank you.
All right. Our next question comes from Bill Kibler at the Altoona Mirror.
Can you hear me?
Yes, we can.
Can you explain how the surveying technology works that enable you to discover the nanobodies, as you said, in the news release, with unrivaled affinity and specificity?
Yeah, so the way our technology is basically based on something called a mass spectrometry, which is a way or a technology where we can use this beautiful analytical tools for the analysis of proteins such as nanobody. The way how these things work is that we will immunize or injected llama wally with the SARS-CoV-2 spike protein, and we wait for example, 40 to 50 days for the affinity to mature. That the llama will produce a high affinity anti-COVID basically nanobodies, and then we use the mass spectrometry proteomic technology pipeline that we've developed and perfected for the past 3 years to quickly isolate these nanobodies in the circulation of the immunized llama wally, and then we can use mass spectrometry to basically determine the amino acid composition of these nanobodies. Once we identify the sequence of these nanobodies, and then we can very quickly produce them as a short DNA, recombinant DNA from microbes such as E. coli cells and E cells, and to quickly and literally produce a large quantity of these nanobodies. Thank you.
All right. Our next question comes from Liz Kilmer at WPXI TV.
Muted. Hey, are you guys able to hear me?
Yes, we are.
Okay, I apologize if this was something that was addressed. I've been having some audio issues on the Zoom, but I understand that the YouTube feed our folks can adequately hear, so I'll be able to play this back. But this morning, Dr. Levine discussed vaccines and plans to distribute. And if you could just kind of speak generally to explain the difference between, this is a treatment versus a vaccine, but can you speak to how the two may go hand in hand when it comes to, mitigating this pandemic?
Yeah, that's a really good question. So what you need to think about whenever we consider this emerging infectious disease is that it's really good to have multiple ways, multiple interventions in development. As we talked about earlier on, it just takes time to get these things through what's typically called the pipeline into a position where they can be used therapeutically. And because we've moved pretty quickly with vaccine development, much faster than normal. Typically, it takes 10.5 years to make a vaccine. And what you're hearing is potentially something might be licensed before the end of this year. Now, we're coming close, of course, to the end of this year. But we would like to think in the next couple of months that a vaccine could be licensed. Of course, that has to be distributed, it has to be manufactured, it has to be produced in millions and millions, hundreds of millions of doses. So that's one part of it. We don't as yet know if those vaccines are efficacious. Efficacious means, do they work? So we have to wait and see, even though we're pushing them forward quickly, do they work? Second thing that we don't know is how long the immunity will last. And that's what's exciting, I think, about having novel other options to treat and prevent. So as has been really nicely described, these these nanobodies are really particular. They're very small, they're easy to manufacture. And what we then need to do at the moment, to answer, even just to speak to some of the previous questions, is we need to ask, can they be used as a treatment? So if you're really sick, can they be used and administered? And again, speaking to how you might administer them, because they're small, they can be blown in the air. If they can be blown in the air, we can get them right into the parts of the body where the virus is most active. That's in the upper and the lower respiratory tract. So can they be used as therapeutic? Or even if you think rather creatively, and again, this is why the preclinical types of studies that we will do in animals matter, can we use them before you might meet the virus? So that would be like being armed, ready to go. Imagine if you have the nanobodies, imagine if you spray the nanobodies, imagine if those nanobodies, which are really potent, are here in your upper respiratory tract. And if you just so happen to touch a door where there's the virus and then touch around your mouth, your mucosal surfaces, they would be ready, aimed, and able to neutralize that infection. Remember, we stop those small amounts of virus at the start, then we mitigate, we reduce the disease, and we protect the individual. So this is what's really interesting about these. There are many ways to use them. It's just not one way.
All right, at this point, I'll reiterate that to ask a question, you'll have to go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in the queue. I will then read your name and your line will be unmuted for you to ask your question. We'll try to call on as many media outlets as we can in the time allotted. In the meantime, I have an email question. Since nanobodies come from llamas, is it possible that people will have a bad reaction to them? Do you think it would be difficult to receive regulatory approval for use of a llama nanobody in humans?
Yeah, and that's a fantastic question. So I would say that if you look at the structure and the sequence of nanobody, realize that the sequence similarity between human monoclonal antibody and nanobodies are very close, both sequence and structure-wise. And indeed, there was the first nanobody drug has been recently approved by EMA and now FDA. On the other hand, I think the ultra potency of the nanobody indicates that we can just apply a minute amount of nanobody directly if it works in humans, of course. And that means that we all know that the toxicity of the antibody drugs is correlated with the amount of dosing. So with a minute amount of dosing, potentially it will be less toxic. But of course, we need preclinical evaluations and especially the clinical evaluations to make sure that this is a safe therapeutics before we move on forward. Thank you.
All right. Our next question comes from Bill Kibler at the Altoona Mirror.
Thank you. Would this, if this were available now, would it be the most effective therapy available anywhere?
Yeah, it's a very tough question, but it's really hard to answer. But I will say that based on the in vitro studies, one of the our best neutralizing COVID nanobodies represent the most potent biologics to date in fighting against the virus. But again, we need to seriously investigating their properties in vivo to make sure that they will be functional. All right, thank you.
All right, our next question comes from Paul Goff at the Pittsburgh Business Times.
Hi, thanks again. Two questions. One, how does this compare in size to, I think it's AB8, right? The monoclonal antibody development at UPMC. And then also, how many llamas would it take for this to become, mass produced? You wouldn't just have the one, right? You need to develop it in a bunch of them. Is that, I know that seems like a long kind of thing, but just wondering about that.
Yeah, I mean, in terms of the comparison between our nanobodies and the AB81 that is developed by from another group, I think there's in terms of size they're highly similar, and it's really nice to see that scientists come up with different strategies to try to tackle the very same problem. In terms of your second question, which is how many animals, how many llamas do we need in order to produce these nanobodies, right? So I think for discovery, because we have a very sensitive technology pipeline, we just need one animal where we can discover many thousands of very potent antiviral nanobodies. As long as we discover the sequence, the amino acid composition of these nanobodies, then we can quickly produce this as recombinant DNA and produce as recombinant protein from the microbes. So we don't need the llama any longer, as long as we discover the sequence. The key is actually the amino acid sequence of these nanobodies. Thank you.
Our next question comes from Aparna Zalani at CBS News Dallas.
Real quick, how is Wally doing? Is he okay or she?
Yeah, I mean, that's a very interesting question. Wally is happy and running around. So last time I asked the farm to send us a picture, I can tell that he's very happy. Yeah, thanks.
And I think what's important to remember is that, speaking back to Paul's question, this is not an animal that is required for production of the nanobody. The real key here is that animal's genetic composition, which produces that one nanobody, is enough then to take into the laboratory, completely independent of the llama that can live happily in Massachusetts. And the laboratory takes over, the bacteria take over, and the bacteria mass produce the nanobody, not the llama.
Yeah, and also, and the other thing is, I think the llama is actually pretty healthy and happy because we're not literally killing the animal. We just draw blood after immunization of 2 to 3 months. So it's perfectly fine. Okay.
All right, we don't have any hands raised, so I'm going to go to another email question. How do you envision this being used? Would we all have inhalers that we would puff on before we go out in public in order to protect ourselves from COVID?
Yeah, that's a fantastic question. I think there are two different possible ways to use these nanobodies. One is used as a prophylactic, that one could do nasal spray to protect, for example, the upper respiratory tract. The other way is actually for early stage treatment, because the key is actually not to wait until the disease progress into the severe disease status, which is very difficult for the treatment, but to basically prevent the disease progression from mild to severe disease status. So I think in this particular case, it will be very nice to aerosolize and inhale deliver the nanobody therapeutics candidates directly to the lung for the potential treatment. Thank you.
We have time for one last question. If there are any participants that would like to raise their hand, or if not, that concludes. Oh.
Sure.
Can you explain it?
Oh yeah, of course. Showing here is basically the virus spike protein. You can see that the beautiful virus spike structure. And our nanobody, this is one of the lead compound, nanobody 21. Basically, we engineer as a trimer just to increase the efficacy of the drug. And you can see that this nanobody 21 trimer in purple sits on top of the virus spike to basically prevent the virus spike from opening, and which is important for the virus to bind to the host receptor and basically for the infection. So this is how this nanobody function. Thank you.
And that concludes the time that we have allotted. Thank you all for attending. In about 45 minutes, we will email media attendees a link to download high-definition footage of this entire press briefing, which we encourage you to use in your news reports. Reporters with follow-up questions are encouraged to contact Media Relations for Pitt Health Sciences and UPMC at upmc.com/media. Please follow @PittTweet and @UPMC News on Twitter for updates. Thank you.